Hi everyone, this is my poster presentation for the LOV fly experiment we did earlier in the semester. The title of my poster is Testing the Neurological Effects of the HTT Huntington's Disease Gene in Drosophila using the LOV GAL4 UAS PolyQ system. Huntington's disease is a hereditary autosomal dominant disorder that arises from mutations in the HTT gene. These mutations consist of several CAG repeats. The number of CAG repeats determines the severity of the disease as well as the risk to future generations because it corresponds to the intensity of the aggregation of glutamines. Less than 28 repeats is considered normal. The next generation is at risk if an individual has 29 to 34 repeats. 35 to 39 repeats means that some people are going to develop the disease, and the next generation is at risk. And those who have more than 40 repeats are going to develop Huntington's, and so will their offspring. Currently, around 30,000 people in the United States suffer from Huntington's disease, and about 200,000 are at risk. The organism Drosophila was chosen for this experiment because its neurological network functions in a similar way to that of humans, as well as the fact that they breed very quickly. Drosophila have vesicles, synapses, and neurotransmitters. The similarity in neuronal networks means that the flies will have phenotypes that closely resemble those of Huntington's disease in humans. We were able to control the expression of the HTT gene by using the LOV GAL4 UAS PolyQ system, and using those crosses, we were able to create an experiment or behavioral assay to analyze the behavioral effects of Huntington's on flies with 128 PolyQ repeats and flies with 16 PolyQ repeats, and compare it to a normal healthy fly that does not express the HTT gene. We conducted this experiment for the purposes of observing the behavioral and neurological changes caused by the expression of the Huntington's gene. In this study, other than the flies, not many materials were utilized. We used male Q128 flies for control, female straight-winged Q128 flies, female straight-winged Q16 flies, 6 vials, and a timer. We separated the genotypes and then isolated 5 flies of each genotype in 3 different vials. Then we taped another vial on top to create a large testing vial. The flies were stunned by repeatedly tapping the vials on the desk. Once stunned, we measured the amount of time it took for the first fly to reach the middle of the vial, or in other words, where the 2 vials met. We stunned and measured each genotype a total of 5 times. Our results came out to be that the control group male Q128 flies took an average of 16.8 seconds. The female Q128 flies took an average of 37.28 seconds, and our female Q16 flies took 51.3 seconds. This was a result we were not expecting. What we expected to see was that the amount of time increased over the course of the 5 trials, and that the female Q128 flies would take longer than the Q16 flies. But this was not the case. In some trials, the experimental flies were faster than the control group, like in trial 4, or instead of increasing, the amount of time it took decreased, like in trial 3 and 4. The one thing that was consistent throughout the experiment was the amount of twitching that occurred. The twitching in the flies was fairly severe, and it was clear that it was affecting their motor function. Some of the flies were not able to just climb up the wall. Oftentimes, they would travel in an upward spiral, stopping several times and for an extended amount of time to twitch, and sometimes they would be nearing the end point, but twitch and jump back down to the bottom of the vial. The stunning essentially intensified the effects and in most cases caused the fly to take longer to climb the vial. One of the things that was most interesting about this study was seeing the differences in each trial. We were expecting the trials to be somewhat similar, but some of them were so drastically different than what we predicted. Studying these flies provided an insight into how and why Huntington's disease research is important, and hopefully one day in the future, scientists are able to develop methods of alleviating symptoms of Huntington's and maybe even a cure for the disease itself. Thank you.